So everyone, hello and welcome. Just first of all, just for you to know, we're going to be answering questions, or I think Harry is going to be answering questions at the end of the session. So if you already have ideas throughout the talk, feel free to write in the chat or just hold to your question and you can raise your hand at the end of the talk. So now I'm going to introduce Harry Lewin. It's my great pleasure. I have been working with him for a couple of years now, and he has a very long CV, and I'm going to tell you a little bit about his academic life. So Harris is a research professor at Arizona State University and a distinguished professor emeritus of evolution and ecology at the University of California, Davis. Until July 2022, he was the Robert and Rosabel Osborne Endowed Chair and distinguished professor at evolution and Ecology. Oh, that's already said. With joint appointments in the Department of Reproduction and Population Health, School of Veterinary Medicine, and John Muir Institute for the Environment. From 2011 until 2016, he served as the UC Davis Vice Chancellor for Research. Before that, Harris spent 27 years at the University of Illinois at Urbana-Champaign, where he held the E.W. and J.M. Gutzel Endowed Professorship in Immunogenetics with a primary appointment in the Department of Animal Sciences. Harris served as a director at the University of Illinois Biotechnology Center, founding director of the W.M. Keck Center for Comparative and Functional Genomics, founding director of the Institute for Genomic Biology, and was a Center for Advanced Study member. Harris' current research interest is in mammalian genome evolution as it relates to adaptation, speciation, and the origins of cancer. In 2017, Harris co-founded the Earth BioGenome Project, or EBP, which aims to sequence all described eukaryotic life, and currently serves as the chair of the EBP Executive Council. Today, Harris will be talking to us about the progress of the EBP, which is a moonshot for biology. And Harris, the floor is yours.
Thanks so much, Camilla. Thanks for the long introduction. It's been a long career, and clearly I have failed at retirement multiple times. So I'm extremely happy and honored to be here. Thank you, Phil. Thank you for the organizers for inviting me to give a talk about the Earth BioGenome Project today. And with that, I'll begin my screen share. Hopefully we can get this all working well. Whoops, we got to go back. There we go. Okay. Okay, so as Camilla mentioned, I'm going to give you an update on the progress of the Earth BioGenome Project and give you a sort of an inside view of where we're headed over the next few years. Okay, so Camilla also mentioned a little bit about the history of the Earth BioGenome Project that I was a co-founder. And so a little more history. Back in 2015, there were several of us who came to obstacles in our own research because we did not have enough genomes to do the kind of work that we were doing. I was working and have been working for more than 25 years on chromosome evolution, which is highly dependent on having contiguous chromosome scale assemblies. My colleague Gene Robinson, I don't know if you can see my pointer over here at the University of Illinois at Urbana-Champaign, is a very prominent entomologist and sort of founder of the field of sociogenomics, and he didn't have enough bee genomes to do the comparative genomics that he wanted to do to unravel the genetics of social behavior in insects. And the gentleman to the far right is John Kress, who is a very well-known plant ecologist and was the Undersecretary for Science at the Smithsonian. And the guy immediately to my, I guess he's to my left there in the picture, is Wanmin Yang, who is the chair of BGI. And together we began to hash out through a series of meetings that were held at the Smithsonian what a project to sequence eukaryotic life might look like. And that project became known as the Earth BioGenome Project. And after about 2.5 years of very intense planning and expanding our group to an international group of very well-known scientists, we published our manifesto in PNAS in April of 2018, which laid out a roadmap and the costs and the reasons for doing the project. And later that year in London, the Earth BioGenome Project was officially launched at the Wellcome Trust, and to some fanfare in Science magazine, The Economist, Nature, all sort of recognized this as a big idea and recognized the importance of doing this conservation. Nature highlighted the work in conservation, but there are many other reasons as well, including bioeconomy, which we'll discuss shortly. So what exactly is the Earth BioGenome Project? It's a confederated international network of networks. It has a common goal of sequencing and annotating the genomes of all 1.8 million currently named species of eukaryotes in a 10-year time frame. That's what gives it its grand challenge nature, that we tried to compress this into a 10-year roadmap of how we would do that. And we had calculated in 2018 that could be done for $4.7 billion over the 10-year period. And we'll talk about some of the revised estimates that we have for the cost toward the end of the talk today. And so the goal then is to create this massive digital repository of eukaryotic life that would represent the collective biological intelligence, if you will, of 3.5 billion or so years of the evolution of life on our planet. And this then, please bear with me for a moment here. Oh no, I see, okay. And so why then is this important? Well, all of us know that much of eukaryotic life on our planet is considered to be under threat due to changes in land use, overexploitation, pollution, invasive species, and of course, most recently, due to climate change. And of the more than 157,000 species that have been evaluated, more than 44,000 species have undergone dramatic reductions in population size and are threatened with extinction. So that's 28% of all assessed species, and most of those are plants and animals. We don't know very much about the protists and some other taxa that are very difficult to assess. But of those that have been assessed, 28% are under some threat of extinction. And it has been estimated that by the end of the century, that up to 1 million species may be lost. And so the extinction rates today are 100 to 1,000-fold greater than the background extinction rates as determined from the geological record. And of course, this has caused much alarm. And now it is widely recognized that the climate crisis is very closely linked to the crisis in existential crisis, really, in the loss of biodiversity. And there are many efforts underway now to try and minimize and stem the loss of biodiversity. I won't talk about that much, but just to make the point that these two, climate change and biodiversity, are very, very tightly linked. And the reason for that is that, the reason that there's such concern is that biodiversity provides the much needed support for all of the ecosystem services that we're accustomed to as humans on the planet, including the provision of fresh water, of food, of medicines from nature, raw materials for building, and for the bioeconomy as a whole. And the current global bioeconomy, the value is estimated at about $4 trillion a year, and that is estimated to be increasing at a rate that it will be at up to $30 trillion by mid-century. So there's a lot riding on the preservation of biodiversity, because if we do not know, we lose 1 million species, what the consequences of that loss will be to these very important ecosystems that are necessary for sustaining life and human life on the planet. And we don't know where the tipping points are. This is, for those of you who are tuning in from Australia, there's a picture from the Great Barrier Reef. And of course, when the reefs die, so goes the entire coral reef ecosystem that is part of it. And that is very important, of course, to humans as well, as these are important breeding grounds for fish that are necessary or that are used in human diets, and many of the economies that are built up around coastal regions. So we simply don't know what the impact of all these losses might be, but they certainly might pose a threat to global stability and our own potential survival as a species. So if all of this is so critical, you would think that we would know most of the species on our planet. And in fact, studies have shown that we probably only have named about 20% of the species on the planet. There are probably up to maybe 10 million eukaryotic species, and only about 1.8 million of those have been named over the past few hundred years since Linnaeus. And bacteria is a completely different situation. We really don't know at all. It could be 4 million, it could be 4 trillion of bacteria and archaea. We don't know. And I might just point out here that there are other initiatives in the world that are dealing with sequencing of microbial diversity. So if we only know about 20% or only 20% of them have been named, how many of those species have been sequenced? If sequencing has become such an important technology, genomics has become such an important technology for conservation, you would think that there would be lots of genomes that are sequenced. Well, of those approximately 1.82 million species shown on this rainbow plot, about 18,000 distinct eukaryotic taxa have whole genome sequences in the INSDC databases as of yesterday, when I checked and made this graph off of the Genomes on a Tree website, which is the main database and resource for genomic information for the Earth BioGenome Project. So only 1% of described eukaryotic sequences have been sequenced. So we're going into this battle really with our hands tied behind our back. In fact, you can go up, through the Linnaean classifications, and you can see that we don't even have a representative sequence for all of the phyla yet. There are still 12 eukaryotic phyla that do not have a representation. So we're at the very, very beginning. And if you look at these 18,000 species that have whole genome sequence, and you break them down by the quality of the assemblies, only 20% of those, so 1 in 5, are of the quality with which we can do the best possible analysis that we can do on those genomes. And that is a really limiting factor, and that means 80% of those 18,000 will probably need to be done over again in order for us to obtain all the information that is going to be needed for conservation, synthetic biology, and other types of applications that we know where very high quality genomes are useful and necessary. So that being the case, well, if you are looking at species that are under threat, there are, of those 44,000 species that I mentioned, about 13,000 are listed as conservation dependent. These are dependent on some human activity to keep their either habitats going or to keep these animals going in botanical gardens or in zoos. And as you can see, less than 3% of endangered species have sequence information. So we're not doing much better. We have 1% of species, but less than 3% of those that are endangered. And so we think this is a real limitation in our ability to use genomics for conservation genomics and for preserving animals in their natural habitats. And so we're not only trying then with this project to aid conservation efforts, but we're also trying then with this comprehensive digital repository to save and to catalog all of the world's eukaryotic DNA, so that in the future, if that DNA is lost, we have a permanent legacy and a record that can be used perhaps for purposes of resurrection of extinct species and reconstitution in the future. Okay, so how is the project organized? I mentioned earlier that the Earth BioGenome Project is a confederated international network of networks. Each of these 58 affiliated projects, many of them are also networks, which means that they are network of networks. The 58 affiliated projects are based in 18 countries and have more than 1,600 participants. As you can see here over on the side, the project is arranged and is organized as a hub and spokes model, where these 58 different affiliated projects are represented by the red circles in this conceptual map. And now we're moving this year toward also adding on the regional nodes, which are shown in gray here. These would be larger aggregations that would include major centers for sequencing and for informatics and other functions, ethics, legal, and social issues, and so on, that may be specific to individual projects or regions. Now, the Executive Council that I chair is part of this overall coordinating function. The coordinating function is housed in the EBP Secretariat that is currently located at Arizona State University. And the coordinating council is responsible for the development of standards for the project. Now, The EBP has 6 different committees that are involved in the development of standards and policies. The International Scientific Committee is, let me see if I can get a pointer here, is chaired by Federica De Palma. And we also have committees, and I'll talk about the International Scientific Committee on the next slide, a little bit more about that. We also have an LC committee chaired by Deborah Matthews, co-chaired by Melissa Goldstein, a JEDI, Justice, Equity, Diversity, and Inclusion Committee, co-chaired by Sadie Paez, Ed Rockefeller, Marcelo Iana da Silva, and Ann McCartney. A communications committee chaired by Nicolette Caparello, who's the executive director at the Secretariat. A nominations committee chaired by Shustin Lindblad-Toh at MIT and Broad, and a chair of the overall governance committee, Rob Waterhouse, who's also one of the chairs of the big affiliated projects and probably soon to be a regional node. That's the ERGA, or the European Reference Genome Alliance. And you can go to the EBP website and you can see all of these, all of the reports on assembly standards. There's going to be a new one coming soon. There's a report on annotation standards, IT and informatics, and a report coming soon on intellectual property. All the reports are there. They're very important, and they are now serving as a basis for many others, both inside and outside the project, as others aspire to produce sequences that are that meet the overall EBP quality standards. I'll talk a little bit about that too. The International Science Committee, again, it's chaired by Federica De Palma, who's at Genome Canada. And we have committees for sample collection and processing, sequencing and assembly, annotation, data analysis, and informatics. And all of these committees are constantly working and updating their reports and standards for the project. And all the projects, the big projects themselves, are trying to integrate those standards into the standards for and governance for the individual affiliated projects. And all of this is overseen by an executive council, as Camilla mentioned. I chair as the elected chair of the council, and there are 8 other members of the council from 7 different countries, and together we try and manage all of this on a volunteer basis. Okay, so what has the Earth BioGenome Project achieved in the last few years? Check that off. This rainbow plot, again, shows it's very similar to the plot you saw before, except the contributions of the Earth BioGenome Project affiliated project are included in the rainbow and would be a subset of those that you saw previously. And so to date, the project has produced genome sequences for nearly 3,000 different species, representing more than 2,200 different genera and over 1,000 eukaryotic families. You might recall from the Earth BioGenome Project manifesto, the Sequencing Life for the Future of Life, that we had divided the project into 3 phases, one which would be sequencing of a representative species from each family, then a representative species from each genus, and then a representative species individual from each of the remaining species. And we have revised our approach because it didn't really make sense to stop or not to sequence more than families when we started. There were many reasons why individual species and subspecies should be sequenced. And so now we have a plan to do this in an overlapping way, about 50% of all the families in phase 1, and then 50% of all families in all genera in phase 2, and then the rest of the species. And where we're at today with that, with the 3,000 species approximately, is we are at, for phase one, we're at about 20% though. So if we figure 50% of the families, we have 10% of the families sequenced in phase one. So we're about 20% of the way there to complete phase one. And as you can see, again, species numbers are quite low, but in the case of the Earth BioGenome Project, these are much higher quality sequences than the typical kinds of draft and highly fragmented sequences that have been produced before really 2018, 2017. All of these sequences can be found under NCBI umbrella bioproject, this PRJNA 533106. You can search that and you can find all of the genomes in the public domain. These are all in INSTC databases, all made public. And that brings up an important point, is that there's much more sequencing going on in the world, and we have not yet captured all of the affiliated projects into the GOAT system. And many projects have been sort of bankrolling genomes and have not yet put them into the INSTC databases. And so the actual contribution is quite a bit higher. We know that, for example, one of the projects that does fungal genomes has done two representative species of each of the 100 approximately fungal families. So that increases these numbers, quite a bit. So still an underrepresentation, and we're working on integrating all of those into the GOAT system. And just to show you how this has been progressing year over year, and also as a function of quality, as you can see, we have been about doubling the last few years in terms of the number of species that are being sequenced by the affiliated projects. And this side of the figure is the number of families, also going up in a linear fashion. But the important point to note from this slide is the ratio of chromosome scale assemblies shown in this teal color to scaffold and contig assemblies. As you can see, the EBP is done really well at switching over from the early years of these scaffold-based assemblies now to these chromosome scale assemblies that are so useful to the scientific community, and making them broadly available to everyone. Whoops. Okay, and something I am extremely proud of, even though my lab hasn't generated really a single sequence, is this are these donut plots, which show you the contribution of the EBP, which is shown in the darker purple here, to the rest of the world in terms of the number of reference quality genomes that have been produced. So I mentioned about 20% of the 18,000 genomes in INSTC, about 20% of those are reference quality. That's what's shown here by 3636. And more than half of those have been produced that meet the EBP quality metrics. More than half of those have been produced by the EBP affiliated projects. And if you go from genera here to the genera, it's nearly 60%, and a really interesting figure showing that we have been targeting families is that 68% of all of the reference quality representative species have been sequenced by EBP affiliated projects. And the most simple metrics, there's more, but the most simple standard for those of you who are interested in this is a contig N50 of at least 1 million. That's for the larger genomes. Could be this 1 million, could be chromosome scale. So it's either chromosome scale or 1 megabase, and a scaffold N50 of greater than 10 million bases, which with our scaffolding techniques, high C-based scaffolding techniques, are now yielding very high quality chromosome scale assemblies in a very reliable and reproducible way. Okay. And they say, well, who's producing all these genomes? And the answer is, though, even though we have 58 affiliated projects, not all of them are involved in sequencing. Many of them are involved in collecting and provisioning samples to larger sequencing labs, and many of them are just getting going. Some of them are transitioning from short reads to long reads. And so today there are really two centers in the world. One is at the Wellcome Sanger Institute, a program called the Tree of Life, which is directed by Mark Blackster, who many of you may know. The Tree of Life program includes the Darwin Tree of Life, which has already sequenced about 1,300, more than 1,300 genomes. It also includes the Psych project, which has sequenced hundreds of butterflies. These are lepidopterans primarily, and also the Aquatic Symbiosis Project, which is a project funded by the Muir Foundation, and doing symbiosis in marine invertebrates. So those three together are, bring us close to about 2,000 species done at Sanger, and then a large number of vertebrate species being done by the Vertebrate Genomes Project, which is led by Eric Jarvis. And there are about 400 of those that have been complete. And then others, the scale is pretty steep. So there are several projects that have done in the hundreds that are still here, and many of which have yet to be fully updated in the system, but we are getting all this information as we go. It's not important, I want to point out, just to count species. You also need to count bases, and because Darwin Tree of Life is sequencing lots of insect genomes, and Vertebrate Genomes Project sequence vertebrates, these tend to be 2 to 3 times the size of the average insect genome. So if you look at the total number of bases sequenced, you can see that well over a terabase of sequence. This is assembly span, not bases, actual bases sequence. This is the span of the assembly. If you look at the bases sequence, it's about 30-fold that, because we do about 30-fold coverage of long reads for these assemblies. So over a terabase of finished sequence for the Darwin Tree of Life, just for this one program, and over 700 gigabases of sequence produced by the Vertebrate Genomes Project. There are other projects, as I mentioned, that are producing in the hundreds that are focused on particular outcomes. The California Conservation Genomics Project, I mentioned for all of you in North America, and really all of you around the world, because this is a flagship project in the state of California and in the United States, which is directed at using genomic information to improve conservation management through to improve conservation management. And this program is led by Brad Schaefer at UCLA, and they're using a landscape genomics approach where they have a reference genome of about 150 threatened and endangered species, and then they go in and sequence 100 to 150 different individuals across different populations in the state, and to assess genetic diversity in those different locations that can then be used for management purposes. So that's one project. Another project there down under is led by Carolyn Hogg at University of Sydney, which has similar goals and objectives to the CCGP. They're focusing on marsupials and other mammals, and have been making a lot of progress in working with people on the ground to implement conservation plans, as well as to develop national policies for the country. I will add here that there are other projects I have not been able to, you really don't have time to discuss. Of course, Camilla's on the call, and she leads the she's one of the leaders of the European Reference Genome Alliance, that project, which has really a large number of countries, I don't remember exactly how many, 22 countries or so involved at least, or maybe 30 countries involved in that initiative. And they have a very strong conservation focus. And there's, for those of you in the southern part of the southern hemisphere, there's projects in Chile called the 1,000 Chilean Genome Project, which has been making a lot of progress, as well as a major project in Belém at the Vale Institute, which is also focused on biodiversity and conservation of biodiversity in the Amazon. And I fully expect a lot more activity in the Southern Hemisphere, particularly in Latin America, and those highly biodiverse countries around the equator, Colombia, Ecuador, Peru, Brazil. These are all important countries. Many of them have in the neighborhood of 10% or more of the total biodiversity that's been described. And so there's much effort to build capacity in those countries and to have them engaged with the Earth BioGenome Project. Now, in the last few minutes, I'll just talk a little bit about the science. The early years in 2023, the first papers were produced primarily with the short read genomes by the Zoonomia Consortium and the Primate Genomes Project. The Zoonomia Consortium had sequenced about 240 different mammalian species. They had 10 papers in the special issue of Science, ranging from phylogenetics and phylogenomics to much deeper understanding of genetic variation as it relates to human diseases. Single base resolution of evolutionary constraint was produced by sequencing these genomes, even though these genomes were not, chromosome scale. They were accurate enough for many, many detailed and informed studies. The Primate Genomes Project, those groups involved in this project were able to sequence more than half of primate species, 233, I believe, out of about 500 species, and they had a representative of each of the primate families and opened many new windows into human genome evolution as well as the evolution of primates. And some of the things that we can do now, those with a short read fragmented genome, some of the things that we could not do before that we could do now were described in this first paper, a landmark paper from the Vertebrate Genomes Project that had the first 16 genomes, of which 6 of those genomes were bat genomes. And we could show, this was Joanna Damas's work in my lab, as we study chromosome evolution, we were able to show that in 5 of those species within the evolutionary breakpoint regions that separated these synthetic regions in human and even in the carnivore, a carnivore outgroup, the Canadian lynx, in 5 out of the 6 bat species, we were able to identify gene loss or pseudogenization within or closely surrounding this evolutionary breakpoint region. And without these contiguous genomes, these regions that flank the breakpoint regions are particularly difficult to sequence because they're very, very high in repetitive elements. And so having these contiguous genomes allowed us to very accurately assess gene gain and loss. These particular genes that have been lost in these different bat lineages were mostly zinc-fingered genes, and many of those, interestingly enough, were related to immunological function. One application. Another application, again, this is from my lab. It's easy for me to talk about stuff done in my own lab, by Joanna, paper published back in 2022, was the complete and reconstruction of the ancestor of all mammals. And as well as intermediate ancestors all the way from about 180, about 200 million years ago or so until human. And we did this in 3 independent lineages. We also used the sloth, collaborating with Camilla, who provided a fantastic genome for us to use, and cow, 3 different reference genome-based reconstructions. And we can reconstruct all the steps, all the inversions, fissions, and fusions at every step along the way in the evolution of from the ancestral mammalian genome to the human genome. From all of this work, we could determine that the ancestral mammal had 19 autosomes and a pair of sex chromosomes. And all of these genomes, if you look carefully, you would see that the genomes were organized in about 2,500 syntenic segments that had not been broken during the course of evolution. Each one of these, and on average, these segments were about 880 kilobases. And these 2,500 syntenic segments are basically the building blocks of all current extant mammal genomes. And lots of other things in this paper, if you haven't seen it, this beautiful work that Joanna did looking at 3-dimensional structure and gene content and gene density and all different types of sequence features in the breakpoint regions and in the conserved blocks. Another beautiful paper published just last year was by a graduate student of Mark's, Charlotte Wright, on lepidopteran phylogenetics and chromosome evolution, and revealed many absolutely fascinating features in chromosome evolution. Some lineages look like they've even undergone chromothripsis, just so many breakages and combinations, and it could be even just another member of the same genus with these highly rearranged genomes. Just fascinating work, and demonstrates some of the beautiful kinds of work that can only be done with reference quality genomes. So in the last few minutes, I'll say, where are we going? Well, we think we'll be done with the 10,000 genomes for phase 1 in another less than 2 years. We're at 3,000 today, and we're ramping up across the world. And so we had a meeting last year in Lausanne to begin to envision how we could complete 150,000 genomes in 4 years. Now, you can imagine there are many challenges with this. We spent a few days locked in a room to rethink the original strategy that we had and get into more detail on how we would actually do this, because to do it will require 37,500 species per year globally over a 4-year period if we want to stay within the 10-year time frame for the project. And that would translate to 721 genomes a week. That would require us to scale 12-fold from the current output that we have in the world today. So we spent a lot of time thinking about how we would do this. The paper has been submitted, and I hope by the end of the year, early next year, you'll be able to read all about this. But phase 2 will be 150,000 species, of which about half of those will be a representative from each eukaryotic genus. A thing that was very exciting to us, a finding that was very exciting to us at the meeting, was when we did the cost revision versus what we had calculated in 2018, that the cost for phase 2 would actually be 400 million less than the 2018 estimate based on advances in technology. But more importantly, we could do it at 400 million less and have all reference quality genomes for the 150,000 for phase 2. In the 2018 paper, we couldn't contemplate that because the cost of the reference genome was at least 2 to 3-fold higher than what it is today. And so this was very important result, very exciting. And I think everyone at that meeting agreed that it would be important to do reference as many of the genomes as reference quality as we could. And when we penciled out the final cost, which I'll show you in a minute, we also, for the 1.8 million species, we also all agreed that we could project certain cost reductions, and those cost reductions would allow us to sequence all 1.8 million species at reference quality. And we could do that at a cost of $3.9 billion. That's sequencing and annotation. And we also had like a $500 million cost for capacity building in the developing world. But if you take that out, 3.9 billion. And if you compare that to the cost of the Human Genome Project, which was $3 billion if we use 2003 as the base year, that's when the Human Genome Draft was completed. In today's dollars, that's $5.1 billion. So all of eukaryotic life can be sequenced at a cost of about 80% of what it costs to sequence the human genome about 20 years ago. And that's really quite remarkable in a few aspects. First, there have been many more investments in the Human Genome Project, totaling at least $14.5 billion on things like ENCODE and all these additional add-up projects that have been added on. And if you consider the return on the investment, this is a report called the Battelle report. There have been other reports that have revised these estimates down somewhat, but the return on investment for human genome, a single genome, and really a couple of model organism genomes, about $1 trillion. And that's a 65-to-1 return on investment. And nobody really knows what the return on investment. We have some economists that are starting to look at this, but there are problems in agriculture and in synthetic biology that are $1 trillion problems, just in one single problem. And so the return on the investment for the EBP is likely to be many more times over than the Human Genome Project. So of course, we think this is a good idea, and one of the reasons we strongly justify sequencing everything, because we're really not sure what we're going to find, versus just sequencing one particular clade or taxon that we think is important. And so $4.4 billion sounds like a lot of money, but I want to put this in the context of big science, because once you talk about $4 billion over 10 years, okay, globally, man, that's really not that much, maybe $420 million a year for 10 years globally, okay, that starts to sound more reasonable. But then you compare this to the cost of other types of big science, and I'll just use two examples. One is the Webb telescope, which cost $10 billion to build, and compare that to 100 Revio sequences, which you probably, for around list price, if you paid that much, would be about 75 million. That's 1% of the cost of the Webb telescope. Could probably be, in 10 years, could probably sequence all of eukaryotic life on the planet. And so the Webb telescope, of course, and all these radio telescopes give us a picture of the origins and evolution of the universe, and our DNA sequencing machines and our sequences that result from those give us a picture of the origins and evolution of life on Earth. And really, if you think about this and you put it in the context of other big science projects like the Large Hadron Collider, which cost like 4.7 billion to build and 13 billion to discover the Higgs boson. If you put it in that context, we think that the return on investment for the Earth BioGenome Project in terms of usefulness is going to be much, much greater. and likely will be the greatest bargain, I think, in the history of science. And I'll close it there, and I'll thank you all very much for your attention. And I will flip all your pictures back up on my screen. And I think we have about 10 minutes for questions.
That is right. Thanks, Harris. Wonderful talk, as always. Very inspiring. I'm sure there's going to be many questions. There are still already a couple of them in the chat. But I will invite people, also the ones who wrote the questions in the chat, if they want to raise their hands, they can pose the questions themselves, or I will start reading. Okay, so let's start here with Phil Betterham. He said, great seminar. A number of estimates suggest that terrestrial insect populations are reducing at a rate of 1 to 2% per year. Given that insects sit at the base of the pyramid, what is EBP doing to expedite the sequence of insect genomes? Should we have a pyramid of life approach?
Yeah. So first, I'll just point out that the largest number of species that have been sequenced so far are insect species. So, but most of those are clustering in just a few clades. And so we are really at the very beginning. Now, I think, we have taken an agnostic view, Phil, in terms of what we think is important. We've taken this phylogenomic wave strategy where we work our way from families to genus to species, is the way that we could get sort of everyone to buy into it. But the reality is the way you get funding for things is that you're going to work on a problem. You're going to work on, maybe you're going to work on conservation, or you're going to work on a particular insect taxa, like, what Gene does and does, social and use sociality in insects. So the way these things are going to get done is likely in chunks, and it's what the individual projects are able to get funding for, because there's no centralized funding, which is one of the big challenges that we have.
Harris, may I give an example of the BG?
Please.
Yes. So in the Biodiversity Genomics Europe project, we actually, we had different approaches, but now the current one is we subcontracted groups around Europe to go on hotspots of biodiversity and do bioblitzes, where they were sampling everything that was there. And this was mostly arthropods and other invertebrates, and that's probably what we're going to be sequencing. So it's pretty much the base of the pyramid. So, and I believe many other projects are going to have similar approaches as well, because as Harry said, it depends on the funding and the goals, the overall goals of the project. So this is going to happen, at least at some level.
And I'd argue, Phil, that the base, one more comment, that the base of the pyramid is this grab bag of the protists, right? I mean, they're really the base of the pyramid, and we know the least about them. And, and they're also the most technically challenging because they're small. There are many single cell organisms, eukaryotes, that cannot be cultured. And so there's a whole group within the EBP now that is focusing on developing sampling protocols and sequencing standards for the massively polyphyletic protists. And so stay tuned there. They're the base of the pyramid.
Another interesting question here. Marcia Marges is saying, excellent seminar. How do you deal with diversity within species? How many individuals or accessions should be sequenced for each published genome?
Yeah, great question, Camilla. As you know, we grapple with this. So the EBP has said they will do one representative of each species, and we already know that a lot can be garnered from a single genome about the genetic diversity of the individual, at least, and the population from which that individual was drawn. And that is an ongoing effort, looking at runs of homozygosity and looking at overall heterozygosity. So a lot can be learned. We've also said that for all, and this is what is budgeted into EBP phase 2, is that all of the threatened and endangered species will have a reference genome, and that we would sequence 100 individuals from, I think it was 10 in the 2018. I think we went to 100 individuals for each of the threatened endangered species, so that we can get a much better view of genetic diversity within at least those species that are threatened with extinction. And so that's part of the plan, but it's not the plan for everything right now. As you know, if you ask that question, it's getting a lot cheaper to determine genetic diversity through, just short read sequencing. And this can be done, pangenomes has been discussed now in EBP. This can be done at a relatively low cost as compared to what it used to cost just a few years ago.
That's right. And there are some projects affiliated to EBP which are doing this approach, right? They are sequencing the diversity of species, like the California one,
right.
The one in Brazil. And so I think this is happening, but again, it depends on the project. Okay, next question. Rogério Magis is saying, wonderful work and effort, but I think you would be also to be concerned on epigenomes. Any initiative coming out?
Yeah, another great question. Depending on what method you use, the epigenomes can also be analyzed. We did not make it a specific objective of the Earth BioGenome Project to reveal all the epigenetic marks, but it's clearly important for understanding the genotype-phenotype relationship and understanding adaptation. And I think these will be done by individual projects. It may come for free it may come for free as the technologies improve. We know ONT is very good for this, but it's hard to get a lot of DNA, enough DNA to do a good assembly using that in technology for many species. So I think it will be done for a large number of those species, but it's not a specific goal of the project.
Yeah, we were having this conversation the other day within ERGA, and I was wondering if this Earth metabolomic project or something, are they also going to include some methylation analysis? Do you know?
Yeah, I don't get a sense that they will do that for the metabolomics, but there are a couple of parallel projects that have been spawned by the EBP that we're working with, and I'm sure there will be a big epigenomics group that will emerge that will use the reference genomes in this way. The group that you mentioned is trying to do metabolomes for as many species on the tree, and a way more complex problem than we have with sequencing, just sequencing and annotation. And then there's the single cell atlas project that you know about, Camilla, the one that's out of Barcelona. And that's another one that very well may look at epigenetic marks and will be very important for the kinds of questions that they're asking in terms of cell fate and multicellular evolution.
Okay, and we have a last question here from Sarah Elgin. You mentioned not only sequencing, but annotation. Will annotation go beyond the computer approach?
Yeah, well, the annotation that we're doing, at least through phase 2, is RNA-seq based, Sarah. And so we will be doing RNA-seq, and so it's computational, but it's not without evidence of transcription. So the transcriptomics will be done in phase 2. And then I think we're all thinking and hoping that AI and other, and other computational tools will be much better by that time at telling us where the genes are and what they are.
Could I also mention that we've had great success in engaging undergraduates in annotation projects. They can learn to do that pretty well. They learn a lot about genes and genomes, and they can do very careful work, particularly if you've got that RNA-seq data. It would be worth thinking about if there isn't any other project underway to engage the undergraduates in this process.
Yeah, it's a great way to engage undergraduates. As Camilla mentioned, the BioBlitzes is another way to engage undergraduates. And as the techniques become more standardized, I mean, what we really want to be sure, Sarah, is that when the annotations are done, they're done in a very, very standardized way. And, right now, places like, EBI and NCBI have their own standard annotation pipelines, and they won't take an annotation that's done by the community. They're going to want a lot of evidence before they accept an annotation done by a community. But we're working toward an organization which would be like a trusted providers, where they are soon going to make their annotation pipeline publicly available because they just simply cannot handle the number of genomes. So if your university incorporates the NCBI pipeline, then there would be no reason why, why those annotations could not be used.
Yeah, I actually wanted to add that at the annotation committee in ERGA, the group has been discussing a lot about non-automated annotation, because this annotation for stuff that is not well standardized, and this include a lot of input from the community. And if you go to some of the material of our annotation committee, there is even the pipeline for community annotation and all this conversation about how to incorporate this into the public repositories. Not that it's going to be easy, but yeah, that's definitely necessary, especially when it's not easy to standardize the annotation process. Are you taking another question?
I'll take another question or two. Yeah, I got to catch a flight in an hour, so I got to get rolling.
Sarah, would you like to say something else?
Oh, I was just going to say, keep thinking in terms of the power of massively parallel undergraduates. You can disperse a standard protocol with the internet, and I think it's well worthwhile to be thinking about how to incorporate outreach projects in the educational sphere in a project of this type and size. That's all.
Well, this is something great for an NSF grant, Sarah, and scaling annotation using undergraduates. I think it's a great idea.
Okay, so last question now from Sibeli Sotero-Caio. Hi, Harris. Thank you for the great talk. Can you comment on how smaller labs could collaborate with these large initiatives? Could a lab, for instance, contribute the RNA or short-read sequencing for a species listed in the wish list of one of these initiatives and join efforts?
Yeah. Well, hi, Sibeli. First, for all of you, those of you on the call, Sibeli is Brazilian, and she works at the Sanger Institute, and those beautiful figures and a lot of what I talked about about GOAT and integration of data into the GOAT, Sibeli is directly responsible for. So thank you, Sibeli, for being on the call, and thank you for all you've done to contribute to my talk today. To answer your question, Sibeli, I think, I think it's possible that smaller labs, I would say smaller labs that develop a specialty that they could then scale. I think that's really the key. If the steps, it used to be that to do the reference quality genome, there were 5 different methods that we needed to produce to meet the metrics. And now it's 2. So one is you need to have the Revios or the ONT machines to do the long reads. And the other is a specialty. It's Hi-C. And when for the paper that I discussed, Joanna's paper on chromosome evolution, all of the high C reactions and sequencing were done by Erez Lieberman's lab at Baylor. So we used all high C data from one place. And so I think specialization is important. Now, annotation does have specialization. You need to be able to collect high quality RNA, mRNA. You need to be able to make libraries. You need to, you need to be able to sequence, and you need to be able to do it computationally. So, yeah, I think there's room for the small labs, but I think it becomes difficult because you want to standardize quality. And it's much easier to standardize your quality when you're doing it at scale. And the same people and the same persons, the same instruments, or everything is done in the same way, you end up with a much more uniform, product. But as Camilla is demonstrating with ERGA, it's really extraordinary, these expertise that pops up in different places. And ERGA is doing a magnificent job at integrating different labs around Europe and different institutes that have specialization and specialties in different areas. And we saw how that worked with Trey Aliotto the other day and his tools. It was great seminars on Friday afternoons from the Vertebrate Genomes Project and EBP that we run. ERGA presented their scheme and their results so far. So I think there's lots of room for capacity building. It's important to build that capacity in Latin America and in the developing world, because the EBP recognizes that a lot of the sequencing that will be done in those countries. It's going to be very difficult to move samples around and be able to get high quality, high molecular weight DNA and RNA that's necessary to do the sequencing and the annotation. We'd much prefer to transfer that technology to countries where the biodiversity is, and then have them share the data under access and benefit sharing agreements, Nagoya, all of that. But the whole focus is to get more countries involved. It's the only way we're going to achieve the scale that's going to be necessary to sequence all eukaryotic species in the world.
Very nice. Yeah, I completely agree with you, Harris. I think there is a lot of space for smaller labs, especially if they specialize on certain taxa, for example, or a specific method. But we need to think it's can be very complicated to coordinate all of this. So this coordination needs to be very well thought, but it's to work with a high number of labs, and possible though, and might be needed, but it's something to consider.
And I'll say ERGA and the Africa BioGenome Project too is very well coordinated what's happening there. And those are two big regional multinational projects that have obtained, funding for coordination efforts, and they're doing a magnificent job, and they're really leading the way in showing how this can be done at a global scale.
Yeah, and Eduardo Ezerek is mentioning here the smaller labs can actually do the analysis. They can do the comparative analysis, the population analysis. They can do a lot. That is true. Yes.
Well, the whole great to see you. I mean, the whole point is to put it out there so like folks like Eduardo and labs anywhere can do the analysis. Free. They don't have to reference, they cite the paper, the genome note, and they can be off and running and doing their own analyses on these genomes, which is just fantastic. I think it's one of the great strengths of the EBP model. Eduardo, go ahead.
Yes. No, good to see you. I agree. That's my suggestion.
Yeah.
Okay, Harris. So I think
that's where the value is.
You can now say goodbye. I think you need to catch a flight. So thank you so much.
Thanks, Harris.
Again, thank you, everyone.
Thanks, Eduardo. Good to see all of you.
Good to see you.
Thanks, everyone, for attending today.